Hello students, welcome back. Today we are going to discuss about NCERT 9th Standard Chapter 5 The Fundamental Units of Life. Today we are going to study about cell organelles. What is cell organelles? We know that there is a division of labor in multicellular organisms such as human beings. This means that different parts of the human body perform different functions. For example, human body has a heart to pump blood, stomach for digestion. This kind of division of labor is also seen within a single cell. Each cell got a certain specific components or tiny organs within it, which is known as cell organelles. Each cell organelle performs a specific function, such as making new material in the cell, clearing up the waste material from the cell, and so on. A cell is able to perform all its functions because of these organelles. It is interesting that all cells are found to have same organelles no matter what their function or what organism they are found in. And these cell organelles are doing a lot of chemical activities to support the complicated structure and function of multicellular organisms. Especially in eukaryotic cell, these cell organelles are membrane bound, distinguish them from the prokaryotic cells. Important cell organelles are found in a cell: Endoplasmic Reticulum, Ribosomes, Golgi apparatus, Lysosomes, Mitochondria, Plastids, Vacuoles, Centrosome. They are important because they carry out very crucial functions in cells. The first one is Endoplasmic Reticulum, as a large network of membrane-bound tubes and sheets, which can be seen as long tubules, round and vehicle type. It may be connected to both the outer nuclear membrane as well as the plasma membrane. Membrane of endoplasmic reticulum, which is very similar to the membrane of plasma membrane, which is present in all eukaryotes but absent in prokaryotes and mature mammalian RBCs. Based upon the presence or absence of ribosome in the surface of endoplasmic reticulum, which is again divided into two, rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum, which is seen as flat sacs or cisternae, which surface is rough because of the presence of ribosomes. It is the main site of protein synthesis, which also synthesize enzymes. Proteins sent to the different sites in need by endoplasmic reticulum. Smooth endoplasmic reticulum, which is seen as vehicles and tubules, which is smooth because the absence of ribosomes in its surface. It is the main site of lipid synthesis, which is also synthesized hormones. In vertebrates, smooth endoplasmic reticulum of liver cells detoxify many poisons and drugs. Some of the proteins and lipids which is synthesized from the endoplasmic reticulum, which take part in the synthesis of cell membrane, the process is known as membrane biogenesis. What are the functions of endoplasmic reticulum? First one, protein and lipid synthesis. Transport. Serve as a channels for the transport of proteins between the various regions of cytoplasm or between the cytoplasm and the nucleus. Both intracellular and intercellular transport of various materials is done by endoplasmic reticulum, which gives an internal support to the cytoplasm as well as to the cell, which mainly take part in the detoxification of the cell. Ribosomes. Ribosomes are membraneless small granules made up of RNA and proteins and found in both prokaryotic and eukaryotic cells. The formation of ribosomes from the nucleolus and the nucleus. Two types of ribosomes are there: free ribosomes and fixed ribosomes. Free ribosomes are scattered in the cytoplasm. They are many in number. Fixed ribosomes, they are attached to rough endoplasmic reticulum, which is known as the protein factories of the cell. Golgi apparatus, which is otherwise known as Golgi complex or Golgi body. as which is named after a famous scientist, Camillo Golgi, which is a membrane-bound organelle of eukaryotic cell, which is made up of series of flattened, stacked pouches of cisternae, which is also seen as tubes and vesicles in the periphery. It is located in the cytoplasm near the endoplasmic reticulum and the nucleus. One or several Golgi apparatus can be seen in animal cells, but in the plant cells there is hundreds of them. What are the functions of Golgi apparatus? First one is elaboration. Elaboration of the proteins or lipids from the endoplasmic reticulum and the nucleus or the other cell components. Transporting, modifying and packaging of proteins and lipids into vesicles for delivery to the targeted destinations which is secreted or excreted as vesicles. Cell wall synthesis, which synthesize all other components except cellulose. Repair and synthesis of cell membrane is also another function of Golgi apparatus. Formation of lysosomes, which is another cell organelle in the cell. The formation of lysosomes is done by Golgi apparatus. One of the important cell organelle in the cell is lysosomes, a membrane-bound cell organelle that contains digestive enzymes, which is responsible for intracellular digestion and waste disposal. Formation of lysosomes is from Golgi apparatus, but the enzymes present in lysosomes is formed from rough endoplasmic reticulum. How lysosomes are important in cell? What are the functions of the lysosomes? Which are digestion. They are known as the digestive bags of the cell. The digestion of food particles, mainly carbohydrate, proteins, lipids, and nucleic acid, is done by lysosomes. Defense. It repairs the cell membrane and also destruction of the foreign particles like bacteria, virus, allergic food, etc. Cleansing. Remove the dead or worn out the cell organelle which makes the cell always clean and cleansed. Suicidal bags. How they are known as suicidal bags? Because the cell gets damaged, it's burst out its enzymes which can digest their own cell. That's why lysosomes are known as the suicidal bags of the cell. Mitochondria is one of the important cell organelle in the cell which is otherwise known as the powerhouse of the cell in eukaryotic cells. It is a rod-shaped, sausage-shaped, double membrane-bound cell organelle which have one porous outer membrane and a deeply folded inner membrane which form cristae. These folds create a large surface area for energy generating due to the reactions which happens in them, which encloses a matrix which contains DNA and ribosomes. Why they known as the powerhouse of the cell? They contain enzymes necessary for the chemical activities and release high amount of energy in the form of ATP, adenosine triphosphate molecules. ATP is the energy currency of the cell. Body uses this ATP for making new chemical compounds and for mechanical work. What are the functions of mitochondria? First one is respiration. They are the sites of cellular respiration. What is cellular respiration? It is the oxidation of food molecules to produce energy in the form of ATP by various chemical activities present in them. Synthesis. It possesses its own DNA and ribosomes, so it can synthesize its own proteins and enzymes. Energy, which provide energy in the form of ATP, which are using for the mechanical work and for making the new materials for the cell. Centrosome, or otherwise known as central apparatus. It is a small organ present near the nucleus. In animals, centrosome divides before cell division, which form two centrosomes. These two centrosomes forms astral spindle pods. These astral spindle pods plays an important role in the cell division for the disintegration of the chromosomes. Vacuoles. Vacuoles are membrane-bound sacs containing non-living solid and liquid contents. They are small size in animals, but very large sized in plant cells. Mainly, three types of vacuoles are present. One is sap vacuoles, which is seen in plants but in some animals also. They containing a liquid content called cell sap, which is covered by a membrane called tonoplast, which contain sugars, amino acids, proteins, organic acids, etc. They provide turgidity and rigidity of the cells. It is also act as a dump for waste products in plant cells. Food vacuoles is another type of vacuole present in amoeba. These sacs containing ingested food particles fuse with the lysosome to form food vacuoles. Digestion occurs inside these kinds of vacuoles because of the enzymes present in these food vacuoles. Example, amoeba. Amoeba forms these kinds of food vacuoles for the digestion of their ingested food particles. Another type of vacuole is contractile vacuoles, which is mainly present in amoeba, paramecium, and chlamydomonas. These vacuoles collect liquid from the cell, swells up, and burst to release their contents, act as a excretory or osmoregulative organ. That means excess water and waste, whenever it is accumulated in a cell, this contractile vacuole absorb these contents and excrete. What are the functions of vacuoles? Osmoregulation and excretion. Storage. And it's a dump place for cellular waste. Food digestion. It gives turgidity and rigidity. Is also absorb water. One of the most important cell organelle, which is only seen in plant cells, which is known as plastids. Plastids are large disc-like cell organelle found only in plant cells, which are specialized to synthesize and store organic substances. On the basis of the pigment present on them, plastids are divided into two, leucoplasts and chromoplasts. Leucoplasts, they are colorless or white plastids mainly meant for the storage of food in roots and underground stem. Example, potato. Chromoplasts, they are colored plastids divided into two subtypes. One is green colored chromoplast. They are otherwise known as chloroplast because they contain the pigment called chlorophyll, which gives green color for the leaves and other parts of the plant. They are the sites of photosynthesis. They trap solar energy to synthesize food, consume carbon dioxide, and release oxygen. They are known as the kitchen of the cell because of this purpose. They possess both chlorophylls and carotenoids. That means the other colored pigments too. Chromoplasts. Chromoplasts are found in fruits, flowers, roots, and stressed and aging leaves, which are responsible for the distinctive colors of those due to the massive accumulation of the carotenoid pigments. Conversion of chloroplast to chromoplast in ripening is a classic example for that. Chromoplasts synthesize and store pigments such as orange-colored carotene in carrot, yellow xanthophylls in bananas and many flowers. Their color varies depending upon what pigment they contain. In fruits and vegetables, in the initial stage, only chlorophyll is present, but in the ripening stage, degradation of chlorophyll occurs. Then massive accumulation of carotenoids gives colors for the fruits and flowers. Change in the plastid pigments associated with leaf senescence or aging, also due to the loss of green chlorophyll which unmasks the pre-existing carotenoids, otherwise the non-green chromoplast. Some flowering plants, they are no or little carotenoids, but they have different colors which due to the colors present in their vacuoles. Example, purple color of grapes or some vegetables. Anthocyanin is the pigment, it's given that purple color. What are the functions of plastids? First one, colors, which gives colors to fruits and flowers, attract pollinators for seed dispersal. Storage, storage of protein and starch in leucoplast is the main function of the Plastids. Photosynthesis. The chlorophyll traps solar energy to manufacture food through the photosynthesis, also maintain carbon dioxide and oxygen balance in the atmosphere. Synthesis. Like mitochondria, it contains its own DNA, RNA, and ribosomes, so it can synthesize its own proteins. Let's discuss about more examples for plastids. Red color of tomato because of the carotenoid pigment concentration increases during the ripening process. Deep yellow color of mango because of the beta carotenoid accumulation during the ripening. Living organisms are constantly making new cells. They make new cells in order to grow and also to replace all dead cells. This process by which new cells are made is called cell division. Cell division is occurring all the time. Around 2 trillion cell divisions occur in the average human body every day. There are two types of cell division. One is mitosis and other one is meiosis. Mitosis. Mitosis is a process of cell division. Here, the mother cell divides and produces two daughter cells. These daughter cells are identical to each other genetically. In the cell cycle, the division process is known as mitosis, where DNA in the nucleus of the cell gets split into two equal chromosome sets. During the growth and development, an organism's body is populated with the cells because of the process of mitosis. The cell division that majorly happen in your body are because of mitosis. Throughout its entire life, mitosis replaces any worn out and old cells and replace it with a single new cells. For the organisms that are single-celled eukaryotes, the mitotic division is a reproduction form which adds a new individual to its population. What are the main features of mitosis? Two daughter cells are formed from the mother cell in each mitotic cell division. This means every nucleus divides into each cell cycle once. The daughter cells resembles the mother cells in size, shape and chromosomes. The chromosome number is the same in daughter and the mother cells. This is what is known as equational division. Mitosis takes place in plants as well in the somatic organs. This is in the stem tip, root tip, leaf base. Mitosis in plants leads to the growth of its vegetative parts. The entire mitosis process consists of just one division. During the mitosis stage, no recombination or segregation take place. Meiosis. Meiosis is used when it is time for the entire organism to reproduce. It's mainly happen in reproductive cells, but the mitosis is mainly happen in the somatic cells or the body cells. There are two main differences between the mitosis and meiosis. First one, meiosis process has two divisions, but there is only one division in mitosis. When the meiosis is complete, a single cell produces 4 new cells instead of just 2 like in mitosis. The second difference is that the new cells only contain half number of chromosomes compared to its parent cell, but in mitosis there is same number of chromosomes in the daughter cells compared to the parent cell. The second difference is that the life on the earth, it allows new genetic combinations to occur which produces variations in life. Examples of cells that undergo meiosis include the cells used in sexual reproduction called gametes. What is diploids and haploids? The cells produced from mitosis are called diploid because they have two complete sets of chromosomes or the same number of chromosomes as the parent cell. The cells produced from meiosis are called haploids because they have only half number of chromosomes as the original cell. What is the difference between the plant cell and the animal cell? One is the presence of cell wall is only in plant cell, which is absent in animal cell. Plastid is also present in only in plant cell, but which is absent in animal cell. Vacuoles. A single large-sized vacuole is present in plant cells, but in animal cell, a few small vacuoles is present. The position of the nucleus in the plant cell in the peripheral region because of the presence of the large vacuole, but in the animal cell it can be seen mainly in the center. In this part, we have discussed about the different cell organelles present in the cell, mainly endoplasmic reticulum, Golgi apparatus, lysosomes, ribosomes, mitochondria, vacuoles, plastids, and centrosome. And also, we have discussed about few facts about cell division and mitosis and meiosis. And also, we compare the difference between the animal cell and plant cell. Hope all of you understand this part. And this is 9th Standard CPSE Science, Chapter 5, the Fundamental Unit of Life. Thank you. Have a nice day.